So thank you, Reese, for the presentation, and also to Brainbox for the opportunity. And my name is Ignacio, and I am a cognitive scientist based in Madrid, Spain. We are mainly working in cognitive control studies and habitual behavior, so automatic behaviors in humans using imaging and neuromodulation studies. So today I will focus on what is our kind of thinking philosophy behind neuroscientific studies, which is circuits and how these circuits organize behavior, and how to being able to modulate them, because until recently this option was not possible. So I will try to explain you all this path and relevant to my field of research, which is cognitive control. So with no further delay, we start. And my field of my brain interested areas are the prefrontal cortex, of course, and as well some other posterior parietal regions. And this the prefrontal anatomy and the subdivisions are very well known in the literature, and they have been associated to many distinct high order cognitive functions. What I will be mostly covering today is inhibitory control and the link with inferofrontal gyrus, as well as the pre-SMA here in more dorsal position. But there are many other functions in these areas, infinite functions that this prefrontal cortex does, which is flexibility between option A and B to switch between different behavioral options, attentional control for the inferofrontal gyrus as well, to monitor special cues that are relevant for a particular goal, as well as conflict monitoring, involving here the limbic circuit with the cingulate cortex that is also activating or recruiting pre-SMA function. It's also related more in the ventral sections of the prefrontal cortex, a bit of frontal gyrus with reward learning, and also a more complex decision making, which is moral functions in these ventral sections, which are the value-based regions of the brain. So these are, of course, there are very few functions here for more, much more that this huge area covers. But I will try to explain mainly inhibitory control functions and how to study with causality protocols. Of course, the brain is organized in loops, and this is one of the topics of the talk based on circuits. And we are very much interested in the associative circuit that also involves subcortical areas, including basal ganglia, striatum, and it is a closed loop that organizes behavior depending on the contextual cues. I won't go into the details between putamen, globus pallidus, and subthalamic nucleus. They all interact to facilitate or inhibit the ongoing behavior. Given associative cue, given a limbic cue, it will pass through different sections of the loop. But the importance of these concepts are that we want to stimulate or we want to modify behavior with one of the areas in the circuit. So far, TMS can apply pulses, magnetic pulses over the cortex, but now with ultrasound, we can cover pretty much the full circuit. So if we want to stimulate deep structures, we also can nowadays. So these are very exciting moments. I don't have still anything to present on that regard, but the idea is that we can cover the full spectrum and circuits to a given behavior. So cognitive control comes from a lot of literature, and we know a lot from cortical regions and functionality in animal behavior. And the classical studies of electrophysiology have depicted that pre-SMA and SMA are subdivisions of the prefrontal cortex, and they differentially engage with motor area. For example, from M1, the pre-SMAs have little neurons that connect or respond to M1 stimulation, while the SMA has much more. So these two regions are already showing different brotherhood with the main motor output. And here in the physiological response from pre-SMA, the very famous study shows how when animals have to switch from one to another action, a large increase of firing potentials are shown in switch trials when they are correctly switching an action. So these two areas are also important in showing how animals learn new behaviors, and it's very much clear with this graph where pre-SMA has a larger set of neurons engaged with new actions, whereas for those that are well learned is a bit less, whereas the SMA has less engagement in new actions. So learning new actions, cognitive control efforts is engaging more the pre-SMA than the SMA, and also it engages when the movement has to be inhibited. So in here a large firing rate when movement was withheld, when the movement was stopped, compared with when the movement was made. So these are very elegant and classic studies of animal physiology to tell us what are the cortical key players for action control, for cognitive control, and they inform very much the human evidence. These are fMRI studies where we put people inside the scanners, and the brain connectivity and the brain function is very well defined in response inhibition, and it activates a set of consistent areas, including the pre-SMA and bilateral inferior frontal gyrus, as well as subthalamic nucleus and other areas, such as caudate, for example, in this study. The key thing is how these areas interrelate to one another, and these models of connectivity establish the directionality and the causality of these regions when the person is trying to stop a behavior. So cortical areas, pre-SMA and inferior frontal gyrus, relate to each other, and they communicate to the motor area to finally produce or not a behavior. And subcortical area, including the subthalamic nucleus, also modulates by inhibitory inputs into the motor area. So these are very useful to think of how activity maps relate to a direction of the different areas and the causal the causal function of one area in particular. Also, it's important to know these functions, these brain areas, because there are certain diseases that also have problems in controlling behavior, such as Parkinson's disease, which is a dopaminergic disease, where patients lose the ability to switch between actions or to inhibit an ongoing action in a motor movement, for example. It is also true in obsessive compulsive disorder, where patients are engaged in repetitive behaviors such as symmetry or excessive washing, for example. They cannot stop doing it. So a stop mechanism is very critical here, as well as in addiction, for example, that patients don't control their desire towards excessive rewards, such as gambling, such as cocaine, smoke, tobacco, and so forth. So all these share a similar mechanism. So it's important to understand where in the brain this mechanism is executed, which is the causal area and the circuit, and then to maybe try to help these circuits be repaired in these patients. So in a brief introduction, I've been telling you about motor control, switching between actions, inhibition especially. There is also evidence of these areas of conflict and learning in pre-SMA functions, as well as inferior frontal gyrus. But these studies with brain imaging don't provide this causality. So tMS and brain stimulation is a great opportunity to test whether these areas are really important for this function. So we can prove this hypothesis with using tMS. So the brain stimulation field is luckily very wide in techniques nowadays. And we can see with this axis of specificity and invasiveness, how one can be more on one side or the other. And basically the ones that I've been using are tMS and electrical stimulation and static magnetic stimulation, which is a novel portable new technique. They are the least invasive, let's say, but as well the pattern of stimulation and the area of stimulation is quite large. So the effects for a cognitive task are not that specific. This can be resolved with ultrasound stimulation, for example, and where you can reach a specific parts of the circuit with quite precise targeting space. So yeah, depending on the degree of specificity you want to reach for a given study, one or other technique may be more valid, yeah, in a way. So in tMS, which is mainly what I will be talking today, I've been doing studies using single or pair pulse stimulation. You can apply one pulse or very close together paired pulses over the motor area while the person is doing a cognitive task. So we get responses from the hand area relative to a given stimulus, relative to a given learning paradigm, and we see how cortical, the motor cortex is excitable or reducing excitability associated throughout a task, throughout a learning, for example. So this is a very elegant to have an online moment of how the cortex is organizing throughout behavior. And then repetitive stimulation as well to have an offline effect, the long-term effect of a protocol, which is mainly is most used using a theta burst stimulation that can excite or inhibit a given area, associated regions as well. So interconnected regions may also change after the stimulation. So with this in mind, what we can resolve is, well, what is the motor cortex? When is it important in cognitive control? In other words, when is M1 sending outputs during cognitive control? We can answer this with tMS. We can also try to answer what other areas influence over M1 and when as well, and as well using repetitive tMS, mainly what is the critical function of a given area, and as well is an area critical for a particular network function. If you remove or apply offline tMS, such as a virtual lesion, what will change in the network? How will the network modify or compensate when a lost function is occurred? That's also a very a rather elegant and important question nowadays to see how the network adapts to a particular tMS protocol. So relative to the chronometry of the cortical function in cognitive control, we know that it's quite a fast process when somebody sees a stop signal, such as a red light, such as a traffic light, such as a lion or a dangerous stimuli. You stop your ongoing behavior, and at 120 milliseconds, this process is starting, and then the motor cortex resolves the output to produce muscle contraction, a muscle movement, and then some form of delay will finally produce a response. So around 220 milliseconds, people are able to stop in a healthy condition. So we want to know at what specific time this motor area is engaged in this last cortical output to generate a muscle movement. And this has been shown elegantly recently. There are many other studies, not many, but a few other studies doing a similar approach where they have a go and a stop signal right one after the other with varying delays. And you can apply pulses 100 milliseconds after the stop signal and covering up to whatever length of pulses you want to test. More or less, people at the beginning didn't know when was the stop process occurring. So the first study tried one pulse at the 100 milliseconds, then other studies tried later, and then this study has tried a few pulses. So it's theorized that it's occurring around 160 milliseconds. In this study, we see that effectively when the pulses were applied after the stop signal, 120, 40, 60, and 180 milliseconds, the motor area was more significantly raising its activity at 160 milliseconds. But this was for failed inhibition. So failed inhibition and go responses produce an activity similar between them, suggesting that when they fail to stop is similar to a go response. So the process is starting somewhat before this 160 milliseconds, because then they fail to stop. So the process should be starting a bit before. How other regions really interact with the motor area to adapt behavior. So if the motor area is the last one to emit a response, what about the pre-SMA, for example, and the prefrontal cortex? This study tried to test this, and they used a task where they had to switch or stay with the same position, with the same response. So here, the subject will have to respond repeatedly throughout 10-15 trials to the same direction, left or right, and sometimes they have to change, which is more costly and demanding. So applying pulses immediately before the response is occurring in both areas, you get a trace of how the motor area is receiving information from these two regions, associated to a stay trial or a switch trial. So you can get a specific conditions and a specific time points. And this study applied pulses at 3 time points, and for the 2 conditions of the task, when they were switching the response, the right inferior frontal cortex here in the right hemisphere was showing more activity connected to the motor area. It was, in other words, it was inducing a larger excitability change when subjects had to switch the response. However, when the subjects had to stay, not produce any new action, and just continue with the same movement, the pre-SMA was showing a larger excitability, a larger effect on the excitability of motor area. So, and this was coming quite earlier than the switch action. So these 2 areas in this study show elegantly how they can manipulate the motor output by switching or staying at a specific time point. So it's been a while for this study, more than 10 years, but it's been very influential in the field. So another study that I conducted was to test a combination of offline repetitive TMS, so a virtual lesion, in other words, and online TMS. So subjects received theta burst over the right inferior frontal cortex compared to a sham condition, and immediately afterwards they performed the task of go and stop signals, also receiving online pulses over pre-SMA or a control condition. So this was to test how both areas could work together, or rather, if you stimulate and knock out the inferior frontal gyrus, will the behavior be altered on its own, or if you stimulate the inferior frontal gyrus combined with pulses that disturb the pre-SMA, also will affect even more the behavior. So what happened is that when we stimulated with theta bars, the offline protocol in the inferior frontal cortex, here we see the kind of the worst inhibition, the slowest inhibition. The SSRT is the value of the stopping ability. So the longer, the worst inhibition. Here we see that there is an effect, and they are slower after receiving theta burst. But we were hoping that in this condition, when they were also receiving pulses on pre-SMA, even longer stopping latencies will be seen, but this was not the case. However, when they were under the sham theta burst, so the inferior frontal cortex was intact, and they were receiving pulses over the pre-SMA, then the behavior was altered. So our hypotheses were not confirmed. We didn't find that impairing both areas will show the worst behavior, but we confirmed that impairing any given area impairs behavior. So interfering with any given area will impair behavior, rather with theta bars or with single pulses. So both areas are engaged in stopping abilities, and this is one of the take-home messages from this study. So pre-SMA, I've been talking a lot about it, and what's the real causal role? We can stimulate and see changes in behavior. What is it really doing? Is it really active or not? So we can see this with fMRI after the stimulation, and this study elegantly showed with how after receiving excitatory stimulation, the pre-SMA was larger, showing larger activity, but it was accompanied by the expected circuitry. So how causal its role in a given behavior if it's accompanied by the expected circuitry. This study tells us that it's a causal relation, but still the circuitry is helping to produce the behavior. So we have come with a series of studies to know what is the chronometry of some of the cortical areas that participate given in the motor cortex alone or helping the motor cortex to produce adaptive behavior. And we know the possible causal essential role of some areas. I prefer to say that an essential role rather than causal, because we don't really know the causality of an area, because it may be doing something that we are not aware of. We just see changes in bold response in the fMRI, but probably it's still doing something. So the causality is a big word for me, but it's very often used. But the full picture will come from circuitry modulation, so an entire modulation of a given circuitry that will give us a real causality spectrum of a circuit, right? So let's see what can we do with this. So I've shown you an example before of fMRI and showing how after stimulation it becomes highlighted the circuitry. But this is another example it was cited in that slide. And the thing about this study is that after right pre-SMA, well, the network compensated to show a new sort of network, which was more directed into the left hemisphere, so in left pre-SMA and left prefrontal cortex. But how was this modulated in a circuit? Well, we also did effective connectivity analysis, and showing the theta bars model and the sham model, they indeed show differences between the regions. So after knocking out the right pre-SMA, a more predominant left hemisphere activity was seen, and a change also in the directionality and the positiveness of the pre-SMA to motor area was seen. So in the SAM model, there was kind of this inhibition of the motor area from pre-SMA, but after receiving pre-SMA stimulation, this was reversed. So here it was an early model, and there have been more developments and improvements. But here we kind of wanted to test the circuitry changes after stimulation, which is what it's important, I think, to do. And recently, using ultrasound, the spectrum can be covered entirely, as I was saying before. And here, this is an example applied in monkeys, where the researchers stimulated the amygdala or the cingulate regions that are not reachable with TMS. So we are now seeing this sort of results, and how stimulating the amygdala, for example, leads to increments of connectivity with cingulate or prefrontal areas in the brain. So it's this is going to open much more studies to look into cognitive control and its circuitry, for sure. But here comes a novel view of the field, where nowadays one area is not sufficient to say it's causal for a given behavior. There are multi-area, multi-function properties in different functions of the brain, including here, as an example, in language, for example, it has been discovered that most of the brain responds to words. So before, we were thinking a couple of areas were involved, such as Broca, Wernicke, thalamus, and a few more. But now, when subjects were listening to a series of words and phrases, and it was a radio program, it comes highlighted pretty much every part of the brain. It doesn't mean that this is not a circuit, but, well, if one wants to now stimulate a language area to test its causality, it's going to be more complicated, because it's a very wide spread activity. So these sort of results are coming more clear in other domains as well, such as visual search or movement. So if this kind of expansion or fashion turns true, it will be difficult to stimulate a circuit because the full brain is doing the job. But anyway, I wanted to comment on this little idea to maybe debate a bit. So, okay, switch into a different topic, which is the emotional impact on behavior. And of course, I've been talking about prefrontal and associative circuits that engage in cognitive control. But I'm going to also present a bit of a limbic transition to also end with pathological and patient studies. So behavior also has to reach a control level when we are approaching others and when we are engaged in a social interaction, and this comes with a moral value. This is based on a given and specific brain regions and connectivity with parietal cortex, as well as anterior temporal lobe and inferior frontal cortex. So these sort of decisions, these sort of behaviors are very daily, they are very frequent, and experience yourself in walking the street and see somebody asking for money. You may give him money or not, and decide to spend it yourself drinking some cocktails. But this may change dramatically if you are with your kid, or you are with your girlfriend, and somebody that you respect is looking at you. You may then be more social acceptable and provide a more moral decision, right? So these sort of decisions are also very very important to study. And one study that was done with fMRI showed that public decision making, when you are observed doing a moral decision, engage a precise network, which was insula cingulate and temporoparietal junction. So as we are in a neuromodulation seminar, if you want to test causality of the circuit, back in 2018, these regions, the most reachable one was the right temporoparietal junction. So we went and stimulated this region. We stimulated the temporoparietal junction compared to a vertex condition, and then subjects played a task, which is a donation task, and had to decide whether giving money to someone, an entity helping children eat, so it's a ONG, or at the same time they will lose money from themselves. So it's a kind of a altruistic decision. It's helping others by losing some of your own. And these choices were done in public or private, while they were being observed by me, actually. So the behavior shows that being in public or private, they choose more when they were stimulated after the stimulated over the TPJ compared to the vertex, and this was larger under the public conditions. So it replicates what you would expect, that when you are observed, you choose more a moral decision. But what is interesting after the stimulation, so the vertex is the control and the TPJ the active stimulation, we see that there is a race in the general decisions towards the other. So the weight between my self-interest was reduced. Here in this axis is the amount of money you lose. So they are willing to lose more money from themselves at the expense of helping the other. So in other words, we modulated by stimulating this area the altruistic sort of behavior of these people. These were healthy people. So going into a brain pathological condition, so if we want to see it as a limbic state where the limbic condition is disorganized, we selected a behavioral addiction. We were in Parkinson's disease patients due to medication to make them move better. Patients develop this sort of addiction. It's defined as the ability to suppress impulse drive on an urge towards familiar stimuli. So the familiar stimuli they normally respond to is food, sex, excessive shopping, They can also be pathological gamblers, hobbyism, internet use, and so on. And this happens in up to 14% of patients. So it's a good model to test an emotional hyperactive state where they have an excessive desire towards these behaviors. So we want to really test and know better what is the underlying mechanism behind this problem. And to do this, we designed tasks that are done inside the scanner. And this one, we selected hypersexual patients and to use a set of stimuli that were already available. They were basically doing a stop signal task, but we combined with erotic or non-erotic images. This is pretty much a problem occurring more in male patients. So the images were selected with female images from a database. And the behavior, what we find is that the on-medication state, when this is produced by medication, so the on-medication produces the worst or worse SSRTs, worse inhibition levels compared to while off medication. So this is the most interesting behavioral stuff. And what is explained by this effect is that the benefits, they were great in stopping benefits while off medicated. So the correlation with the SSRT were larger when controlling for the off medicated state. So this is an effect driven by medication for sure that is impairing the ability to stop. And in the brain, what we can see when the patients were succeeding to stop in erotic versus non-erotic, there was a pretty large network involving caudate, cingulate, PTA, as well as parietal cortex. And specifically between the groups of patients, they were also replicating some areas, but including also the pre-SMA and cingulate hub. So if we want to know what are the explanations of these areas, what is the circuitry, what is the most important area driving this aberrant behavior, we need to really test with a model, and we selected dynamic causal modeling, where you introduce a series of areas with a time series bold activity and test different directions. We just have to test based on the non-anatomical connectivity. And it turns out that we had a winning model that explained most of the connections. And this winning model included a top-down control from pre-SMA to caudate and cingulate. And specifically between all the controls and the two group patients on and off medication, to make it more clear, the most interesting finding was that patients with the impulsivity, with the hypersexuality, had a significant difference of the local activity in the pre-SMA when the task was seen. So when the erotic stop inhibit stimuli was processed, there was a significant difference between those that did not develop the problem, as well as a descending track difference with the caudate connectivity. This was reduced in patients with hypersexual behavior. So that's pretty nice. It allows us for cognitive scientists and brain stimulation people to say, okay, we can stimulate the pre-SMA because it's more or less a reachable target with TMS. And if we would like to stimulate other areas, that would be more difficult with TMS. So we did that, and we closed the circle in a way because we found altered behavior with the task, define a few regions with a model that seems to explain this regional difference between patients. And we applied TMS over the pre-SMA. We applied real versus sham conditions, so it was a within subject design. And what we find there is that in the real condition, patients are faster than the sham condition in general, but also the inhibitory ability was faster than in the real condition compared to sham. So in other words, we sort of improved the inhibitory ability when exposed to erotic images. This was happening across erotic and non-erotic. We think it can be a general effect of the images and the brain stimulation that has a general global effect in inhibition and also in other type of non-erotic images. But we are quite happy with the result, because we find that at least in an acute manner we can modulate behavior. So this may open a new strategies to try to prolong the effects with more repeated sessions in this hypersexual problem. So I will start to finish with this. The conclusions are that cognitive control is dependent upon Pre-SMA on the inferior frontal cortex. We have also seen, and it can be improved with tMS in healthy controls. We can map cortical hubs with varying methods of tMS, and it's useful to study a certain form of causality, as well as the temporal features of control of behavior. And one region is not essential. We can tell that network changes may explain the new observed behavior. So we need to really specify, after stimulating a region, if imaging data is available, of course, what are the compensatory mechanisms of the network itself. In the domain of emotional and moral decision-making, social functions can be modulated by stimulation, turning people into more altruistic forms of behavior, which is quite nice to pretend. But yeah, of course, this is an experimental context. And then emotional changes bias frontostriatal circuitry. We know that when the patients receive sexual cues and have hypersexuality, but we can remediate that problem with neuromodulation. We can stimulate, and at least shortly with a given task, it does seem to improve the control over this type of cues. And then, as a general wrap-up, cognitive control can be multifactorial. It can operate across domains and action control especially, but as well in moral decision. Probably we didn't test specifically for that, but it can be a control function as well in morality, and in emotional clinical context is also very relevant to study and try to understand better mechanisms behind altered cognitive control in neuropathological conditions. So I finish here, and this is Madrid, and I thank the people involved in my team and collaborators in France and Zurich for the tMS and imaging studies, and also the funding. So thank you very much. I leave it here if there are questions.
Thank you very much, Ignacio. It's nice to see Dr. Fernando Alonso can take some time out of his Formula 1 career to help you do some research as well.
Yeah, that's...
No questions as yet, but I think that's due to a very nicely put and clear, methodology and research.
So I was too clear. I should have been more vague to leave open space.
Some more ambiguity next time, and so of course anyone is welcome to post any questions. I'll kind of start to wrap up from a BrainBox point of view as well, and yeah, by all means, if there are any questions about anything that BrainBox do, or of course any of Dr. Obeso's research, then by all means do ask. So if I just share these with you, could you? I hope those are showing. So just to give you a quick kind of run through of some of the other stuff that that we as BrainBox do. We've been operating as an initiative since 2016. So the initiative kind of encompasses, all of the event side of stuff. So whether that is in the form of a webinar or a workshop or a conference, as you'll see as we go through, it's kind of why we have the initiative to help kind of give back and promote research from the community that we support with the product side of stuff as well. So of course there is the webinars. We are taking a little break from these over the next couple of weeks to focus our efforts on the conference, which is coming up, and then a couple of workshops later in the year as well. But if there is anyone attending who has a specific kind of research area or any of their research they'd really like to promote, then by all means do get in touch with us. We'd love to host these webinars with you. Anything you might be interested in that you may have missed previously, you're able to catch up on those on our YouTube channel. There's a whole host of webinars on there. The workshops then coming up, so those are a chance for anyone who might be new to a technique or just like to understand a little more. It's some really dedicated, it used to be hands-on time, it's now virtual hands-on. We'll be hands-on again in the future someday. But it's a chance for you to have some real kind of, one-to-one time with researchers in the field and with people like me, product specialists in specific equipment, to really kind of get to grips with anything related to the topic, but at a bit of a slower pace. So that, yeah, anyone who's new to any of these areas, again, do get in touch. The conference then, so next week we'll be hosting our, I don't know how many years now, maybe 5 years. Andrew that might be wrong. Sorry if it is. It's been online the last couple of years. Obviously, travel restrictions prevent us from putting on a worldwide conference for everyone. Fingers crossed next year we'll see some of you in person. But yeah, 2021 conference next year. Registrations are still open, £30, seeing as it's an online only conference. We'll be having a whole host of talks about TMS, TES, transcranial focused ultrasound, as well as the imaging that supports all of these research techniques. So fMRI, NIRS, EEG. The speakers over the conference, who's who of brain stimulation and imaging here, as well as obviously the poster sessions, some kind of after conference drinks. You have to provide the drink, unfortunately. But yeah, lots of conversation, lots of networking opportunities available here. And of course, none of this is possible without the other arm to BrainBox, the product arm. So anything to do with brain stimulation, any imaging modalities, they're available from us at BrainBox. So by all means, if anything has kind of piqued your interest today, yeah, go ahead and have a look at brainbox-neuro.com. As well as that, I've finished the slideshow. Any support that you might want to see or you might need, that's available from us as well. So support at brainbox-neuro.com, or me directly is mark@brainbox-neuro.com. So I'll double check the questions again. We have one question. Thank you very much for the presentation. I have a question regarding the pre-SMA target. Did you test the effect of inhibiting the pre-SMA on your other behaviors, for example, motor task performance? Thank you. From Nabila.
Yes, thank you, Nabila. Well, one of the studies presented today, we had a control test for a Go simple task, and it didn't affect any of these parameters. I also have using another technique, which is transcranial static stimulation. We stimulated SMA with a kind of a simpler planification task, and we saw that there were slower movements. So this technique induced a worse performance in a simple planification task, motor planning task. But for these studies, we didn't see effects on that simple go condition. Thank you for your question.
That answers your question, yeah. So with that then, I think
you can say that if there are other questions, I think I left my email somewhere, or yeah, just you can send me a quick email, no problem.